Hello, everyone. Thank you so much for joining us today. I'm Saber, and today I'm going to present my thesis project named DEEMD. For this presentation, I start with an introduction to the problem that we want to solve, and then I explain our proposed pipeline and the experimental setup that we use to test it. And finally, I discuss the results. The COVID-19 global pandemic has urged the research community to focus on SARS-CoV-2 for discovering potential therapeutics. Amongst multiple endeavors, drug repurposing has been a major focus. Drug repurposing aims to identify existing clinically approved treatments that possess effectiveness against another disease that could be redirected towards clinical use. With the advantages such as established pharmacological and safety profiles, as well as a robust supply chain, this approach is especially practical and beneficial in case of an outbreak. Most drug repurposing studies rely on a high throughput screening to enable them to monitor multiple drugs at the same time. One of the rapid approaches for this screening is high throughput morphological analysis of cells stained with multiple fluorescent dyes. Morphological analysis uses the morphological features from single cells or cell populations as an observable variable to draw a conclusion about the underlying cellular dynamics. These features can be extracted from images of infected and uninfected cells and then be applied to images of infected drug-treated cells to predict drug efficacy based on cellular morphology changes. We had two goals in this work. Our first goal is to localize the infected cells within a sample image population without using any specific molecular tools for tracking the virus by training a model that distinguishes between infected and uninfected sample images. These localized cells can be later used for inspection about their biology to better understand the infection. The second goal is to estimate efficacy of treatments based on the morphological features so that we can assess the effectiveness of a library of treatments within a high throughput screening. We hypothesize that an effective treatment causes treated infected cells to look morphologically similar to the uninfected ones. So if we somehow manage to extract morphological features induced by the SARS-CoV-2 infection on the cells, we can use them to come up with a measure that allows us to compare treated infected cells to the healthy ones and quantify how effective a treatment is. But first, to be able to design a model to extract morphological features that we are looking for, we need to understand how an infectious agent spreads. When a sample cell population is infected with a virus, only a portion of the cells are infected. And this portion is mostly controlled by the multiplicity of infection. That is the number of virions that are added per cell during the infection procedure. In the microscopy images of sample cells, there might be no probes or tags for the viruses, especially if the sample were imaged at very early stages of an outbreak, like COVID-19, where those probes are not available. So there are no pixel level annotations that indicates which cells are infected that we can use to extract and learn the morphological features that we are looking for. The only label that we have is whether the sample is infected or not. And the main question is how to identify which cells are infected only using this label. Our proposed method, DEEMD, consists of two components. A component that is able to classify the sample images into uninfected and infected class to learn the morphological features induced by the SARS infection on the cells, and a component to estimate compound efficacy against SARS-CoV-2 based on morphological features learned in the first step. I will now go into the details of these components. We formulate the identification of infected cells within the sample images using multiple instance learning, or MIL. MIL is proposed as a variation of supervised learning for problems with incomplete knowledge about the labels of the samples. In the typical supervised learning, every training sample or instance is assigned with a label. On the other hand, in MIL, the labels are only assigned to sets of instances called bags, and there are no labels for individual instances in the dataset. The goal of MIL Learner is to classify unseen bags or instances based on the labels that it had during its training procedure. With that in mind, we can use this framework to formulate our problem. We can break down the images that we have into smaller patches and think of them as instances. We have samples of infected populations and samples of uninfected populations. And we have two questions that we want to answer here. Given a new sample image, is this sample infected or uninfected? And if it is infected, where are the infected cells? There are multiple assumptions about the relation of the labels and the instances in the MIL. However, the most common assumption that is well suited for our problem is the standard instant assumption. Based on this, we assume that if a sample cell population is infected, then there should be at least one patch that contains infected cells. And an uninfected sample cell population does not have any infected cells. With that being said, we can use a deep neural network as a learner in the MIL context. And here's how the training procedure goes. For each sample image, we break it into smaller overlapping patches. Then each patch is fed into a neural network, a ResNet50 here, for a binary classification between uninfected and infected class. The output of the model is the probability of a patch being infected. Then for each sample image, all of its patches are sorted based on their infection probability, and the k-top patches are selected for training. These patches are assigned with the sample image label that we have in the dataset, and the loss is calculated based on a binary cross entropy function. Then the gradient is calculated for backpropagation and updating the model. The mathematical expression for the loss function helps understanding this procedure better. Here, yi is the label for the IS sample image in the dataset, and mu-I pj is the predicted infection probability for the JS patch in the IS sample image by the model. The loss function is a weighted binary cross entropy function that compares the predicted values with the yi, as you can see here. We then sum up this function value for all of the patches that are in the set of k-top patches with the highest infection probability for the IS sample image and average them. And finally, we take the average over all training sample images and use it to update the weights of the model. Something to mention here is that the ranking and selecting of the k-top patches is invisible to the model and are not included in the backpropagation. They only affect the set of training patches in each iteration that are being used for updating the model. So within each iteration of training, we filter the instances based on their infection probability and use the ones that we have confidence in their labels to train the instance classifier. We repeat this procedure until the loss function has stopped decreasing on the validation set. Once the MIL model is trained, we can use it to extract the features that we need for estimating treatment efficacy scores based on our initial assumption. Our initial assumption about the effectiveness of a treatment is that an effective treatment would prevent drastic infection-induced morphology changes in the cells. Hence, the morphological profile of the treated cells would be similar to that of uninfected ones. Assume that we want to estimate the efficacy score for a treatment TI at concentration CJ. The dataset includes multiple sample images of cells treated with TI at CJ, serving as replicates. We start by breaking down the image into smaller patches and feed it into a MIL model to calculate the estimated patch infection probabilities. Now we have to aggregate the estimated patch infection probabilities to form the sample infection probability. Now we have infection probabilities for all of the replicates of the treatment TI at CJ, and we have to aggregate it furthermore into an efficacy score. Here you can see an example for one of these distributions. Since we do not have any assumption about the distribution of the infection probabilities across replicates, we used a non-parametric statistical test to estimate the median and its confidence interval for this distribution, as shown as a shaded region here. We then use the upper bound of this confidence interval to be more conservative about the false positives and define the efficacy score to be 1 minus the upper bound, estimated upper bound. Essentially, if a treatment sample does not have any infected patches, it shows that the treatment was effective. And if other replicates also exhibit the same results, then the treatment is considered to be effective by the model and gets a high efficacy score close to 1. In order to identify effective concentrations, we compare the respective efficacy score to a threshold. Inspired by the concept of EC50, we choose this threshold to be 0.5. And finally, to form the final ranking of the treatments, we aggregate all of the effective concentrations of the treatments and rank them respectively. To compare the performance of DEEMD, we used two baseline models. The first one is the whole image-based model that is a trained CNN based on ResNet 50 to distinguish between infected and uninfected images. We used full resolution images as input and used the labels associated with each image in a binary cross-entropy loss function, similar to the MIL model. The second model is named patch-based model, which is an extreme case of the MIL model, actually. Instead of limiting the model to only take K top patches in the training, we use all of the patches and label them with the sample level label that we had, or equivalently, we set K to be N. The patch-based models is also based on the ResNet 50, and all models were trained and validated with the same data splits to make it a fair comparison. In order to test DEEMD, we used RX-19 dataset published by the Recurgent Pharmaceuticals. It includes monolayers of normal human renal cortical epithelial cells in 3 viral conditions. MOC, which is the healthy control samples, UV inactivated SARS-CoV-2, which are the samples that were infected with the UV irradiated inactivated virus. And the third group is active SARS-CoV. The active SARS group is further divided into 2 subgroups, untreated set and a set which was treated with a library of FDA-approved compounds in more than 6 half-log concentrations, with 6 replicates per dose for each compound. The samples are then fixed and stained with 5 different dyes, Hoechst, Kane, Floydin, Cyto14, and WGA to detect various subcellular structures. Here you can see an example composite image from the dataset, along with a GIF showing each stain separately. We merged the MOC and UV inactivated class since we did not observe any morphological changes between these 2 groups. The samples are then split into 4 non-overlapping sets: training set with around 20K samples, validation set, untreated test set, and treated test set. The training set is used for training the model, whereas the validation set is used for selecting and tuning the hyperparameters. The performance of the tuned model is measured on the untreated test set, and the treated test set is solely used for estimating the efficacy of treatments. We measured the performance of the models on the untreated test set using precision recall curves, as you can see here. We can see that all models are quite capable of classifying the sample images, and the patch-based model is even slightly better than the other 2, indicating that these models have learned morphological features which could be generalized to unseen untreated test set. However, we need to keep in mind that these models were trained in very different conditions, and their extracted morphological features have different characteristics due to their input and training procedure. The MIL model has learned to extract highly discriminative features from micropopulations or single cells, whereas the whole image-based model focuses on a macropopulation of cells and learns morphological features which are not as detailed as the MILs. The patch-based model lies in between these 2. On one hand, it has been trained using patches with the micropopulation features, but on the other hand, the labels that it was provided for training were noisy. Thus, the model is not as focused as the MIL models. These shifts in the learned feature spaces are further transferred to the downstream analysis of the treatment efficacy scores, as we will see in later slides. Here we see the dose-dependent estimated efficacy score for a sample compound by the MIL model as an example. On the x-axis, we have the log concentrations, and on the y-axis, we have the dose-dependent estimated efficacy scores. The violins show the distribution for the estimated efficacies for the replicates for the same dose, and the blue curve is a logistic curve fit to all of the estimations. The violin is opaque if the model has identified the treatment to be effective at that concentration. If a violin is concentrated at high value, the treatment is more effective at that dose. Here you can see that the treatment was not very effective at low concentrations, but as the concentration increases, the effectiveness also increases. And this is similar to what you expect to see in an effective dose curve with the experimental assays. Here we see the same violin plots for top-ranked treatments identified by the MIL model for all 3 models. You can see that the MIL model of efficacy estimation are in the top panel are structured. As it is expected for an effective antiviral compound, the dose-dependent efficacy score increases with increasing the concentration, similar to the fitted logistic curves. We expect to observe a higher effectiveness with increasing concentration of a treatment for an antiviral which is potent against a specific target, up to the point that it does not introduce toxicity to the cells. On the other hand, if we look at the whole image-based model on the bottom panel, the predictions are scattered randomly among all treatments and lack any form of a structure or pattern. As I mentioned earlier, the patch-based model is a hybrid of the other two models in terms of its training. We can see here in the middle panel that the estimations are slightly structured, but not as much as the MIL model, implying that the model was not able to extract the features quite completely due to its noisy training environment. Despite the fact that these models are capable of accurately distinguishing between infected and uninfected samples, their learned morphological feature spaces are not detailed enough for treatment efficacy estimation. DEEMD has identified treatments that are reported in the literature to possess therapeutic activity against SARS-CoV-2. Here is a table for the identified treatments, along with the studies that have reported them to show some kind of effectiveness against SARS-CoV-2 or COVID-19. We also compared DEEMD estimated efficacy scores with the original paper that published the dataset and used a similar pipeline to estimate the efficacy for the treatments. We can see that the treatments with high efficacy scores, namely remdesivir, GS44, and aloxastatin, were also assigned a high hit score by the original paper. Note that even some of the identified treatments have already gone under clinical value and show to be effective against COVID-19, such as colchicine. Thanks to the special training and inference procedure we have with the MIL, by aggregating the patch infection probabilities, we can come up with an infection map. Notice that in generation of these maps, annotated as red here, no form of annotations was used. Instead, the model uses the sample level labels to localize the infection. Visual inspection of the infection maps, annotated as red again here in these images, suggests that the MIL model is focused on high intensity areas, which are present in all channels except for hoax, as you can see in this example images. We hypothesize that these are dead or damaged cells resulting from cytopathic effects, or CPE. CPE refers to changes in host cell structure as a result of viral infection, and SARS-CoV-2 is known to be a cytopathogenic agent. This suggests that the MIL model has incorporated biologically relevant morphological features into the infection maps, as we expected it to do. DEEMD has its own limitations. The main underlying assumption in any morphological analysis is that the agent is known to introduce morphological changes in the host cell. If this assumption is not met, then the morphological analysis is not applicable in the first place. The second limitation for DEEMD is drug toxicity. Drug toxicity refers to compounds' negative side effects on a living cell. If the treatment has toxic effects or induces other changes in cell morphology, the sample may no longer resemble either the uninfected or infected class, even if the treatment was effective at rescuing the cells. Therefore, in DEEMD, low efficacy score does not necessarily imply that the treatment is ineffective. It might be the case that the treatment has changed the cell morphology to a non-healthy state, and yet the treatment is effective. However, DEEMD would not identify this, and the model's prediction would not be conclusive in this case. In summary, in this work, we present a method capable of estimating treatment efficacy of compounds based on morphological analysis. It includes a deep learning model that's trained within multiple instance learning framework to extract morphological features corresponding to SARS-CoV-2 infection in micropopulations, as well as generating an infection map in a weakly supervised fashion. We compared the performance of the MIL model to two baseline models and demonstrate its effectiveness. DEEMD also identifies compounds that have been suggested by the community to have antiviral effectiveness against SARS-CoV-2 or COVID-19, supporting the performance and validity of the proposed pipeline. And that concludes my presentation. I would like to thank my colleagues and collaborators who helped me in this work, and thank you for your attention.